Hello all, welcome to my channel. In today's video lecture, we will be looking at the various methods for purification of viruses. So we know that viruses can be grown or cultivated in cell cultures or in the live plant or the animal itself or even an embryonated egg. But once we have grown these viruses in this culture medium, now to study them thoroughly, we have to obtain them in a pure form. So for purification of viruses, or when we say pure form, to obtain the viruses from free from any contaminating material such as a cell or the cell extract, we need to apply certain techniques which we are going to look into today. Now, none of these techniques are foolproof and no one technique is considered as the gold standard for purification of viruses. Every lab has their own optimized, standardized technique. But these are some of the basics of the current techniques that are being employed in the industry. And usually a combination of techniques is used so that it gives us better results and purer extracts of the virus. So the four techniques we'll be looking into today are ultracentrifugation, ultrafiltration, precipitation and chromatography. These are the four techniques that we will be looking into. So the first one is ultracentrifugation. It is a specialized technique which is used to spin the samples or move the sample at very high speed. Just like our normal centrifugation, here also the principle is that the components in a solution are going to be separated based on their size and their density, or in not only that, even based on the viscosity of the medium or the density of the medium or the solvent. So if I keep a solution at the room temperature for an extremely long period of time, I'll have the particles settling down due to gravity, because gravity is going to pull them down. But if I add centrifugal force over there, if I spin the particles at a high speed, the centrifugal force is going to increase the rate of sedimentation. So the sedimentation would have occurred naturally due to gravity, but it's very slow and time taking. So what we do is the centrifugal force is going to aid the gravitational pull and increase the rate of sedimentation. Now, how this can be done is by applying very high speeds, spinning the sample at speeds of 10,000 rpm, 40,000 rpm as well. What the rpm here means rotations per minute. So currently we have ultracentrifuges which can spin as much as 1,50,000 rotations per minute or 1,50,000 rpm. In 1 minute, it takes 1,50,000 times spinning. Now, how does this help? What we do here is that the sample containing the virus is spun like this, or it is put into an ultracentrifuge, and at this very high speed it is made to spin so that the particles, the other extracts are going to settle down and we get our viral sample separated. So we get a supernatant, that is the liquid form, the solvent which is present on top, and we get a pellet which is present at the bottom. Now what happens over here is that when we spin at such high speed, such extreme high centrifugal force, that will definitely cause overheating, and that can cause damage to your sample, to the virus. So usually the ultracentrifuges are equipped with vacuum systems which make sure that the temperature is constant inside the centrifuge rotor. The temperature is maintained constantly due to the vacuum. Additionally, there will also be a cooling system. So the presence of a vacuum and the cooling is helping us to use such high speeds without causing any damage to the particle or to the virus. This is called as ultracentrifugation. So what we do here is that from the regular centrifuge, this gives us an additional advantage because the spinning speed is much higher. And viruses being really tiny particles, you need to spin them at such high speeds to obtain them separately, or to separate them out or to differentiate them. Now, the ultracentrifuges have been developed since early 1900s, and we have been using it for the differential separation of various cellular components, not just viruses, but even for organelles or for separating out the lipid membrane, even purifying the DNA and RNA. But there are two major methods of ultracentrifugation which are in use for viruses today, which have been shown over here. The first one is differential centrifugation. Now, this is a technique which is routinely used to separate the components of a solution based on the sedimentation rate. Now, here the different fragments we know are having different size, different densities. So, each fragment will settle into a pellet with different minimum centrifugal force. So meaning to say, at every time, with every spin, consequently we keep increasing the centrifugal field, or we keep increasing the centrifugal force. So the separation of the sample into different layers can be done by centrifugation at different speeds. Now, every time we centrifuge, we are going to get pellet of a particular particle that is present in the solution, which is going to, whose size is going to decrease with every spin. So as you can see over here, first we had the large size green particle settling down, then we had the red colored ones, the blue, and finally we have the black colored ones. Usually viruses are the tiniest particles in a solution, and they will take the longest to settle down. So what we need to do here is that the separation of the sample has to be done into different layers by consequently increasing the centrifugal force. So under weak forces, the pellet will be made up mainly of cells and the larger particles. But as we keep increasing the centrifugal field, each time a portion of different density is going to get separated, or different density is going to get extracted. So repeatedly, this leads, when we do this repeatedly, it leads to, or it produces a rank of layers. Every layer is going to have a different density, and this is how we can, we are stepwise separating out the pellet. So with a stepwise increase of the centrifugal field, we are going to separate out or particles which are smaller and smaller in size. So this is differential centrifugation, a quite easy method of centrifuging and quite easy method of obtaining viruses in their pure form. The second type of ultracentrifugation is the density gradient ultracentrifugation, slightly better one, and it helps to separate out our particles by using their density. So it's not just the size here, we can even separate out particles having similar size but different density. If they have, we can quickly separate them out. Now what we do here is that to aid in the sedimentation, to aid in changing the density, we are going to even use a solvent over here. So we can use viscous liquids such as glycerol or sucrose. They can be added to the sample, they can be mixed with our viral sample. We can even use cesium salt gradients, or any such viscous solution can be added, which will help us to establish a gradient in the density. So based on the density, based on the sedimentation properties of the substance, we are going to separate out the particles. To aid this, we are adding certain solutions such as sucrose or cesium salt, or we can even add glycerol, any kind of viscous liquids. In fact, nowadays we have several commercial gradient kits which have certain solutions in them that are designed for certain or specific particles. So there are two types of density gradient centrifugation. We have rate zonal density gradient centrifugation, and we also have isopycnic centrifugation. Basically what happens is, in both of these, we are isolating or we are extracting the particles based on their density alone. But then the way we are applying the density is what changes. So in short, viruses can be purified, one of the methods being ultracentrifugation, under which we have two broad types: differential ultracentrifugation and density gradient ultracentrifugation. Under density gradient ultracentrifugation, we again have two more subtypes, that is rate zonal and isopycnic. Isopycnic is also called as equilibrium centrifugation. Both these methods are using the centrifugal force. They are based on the size and the density of the viral particles. But the major difference between these two techniques, between differential centrifugation and density gradient centrifugation, is that this method of density gradient is using a particular solution. Like I said, it could be sucrose or glycerol or phycol or cesium chloride or some gel. So we make the viral sample pass through this solution which is having a separate density, so that the density of the particles in the solution will vary. As we are passing them through the solution is going to separate the sample into layers by relative density. So every time we are centrifuging, the molecules are settling down under the centrifugal force till they reach the density which is same as that of the medium. Till then, they'll keep settling down. Once the medium's density and the particle's density is same, they'll stop settling down. So this is the idea behind density gradient centrifugation. But in case of The differential centrifugation, we don't add any extra material or solvent over there. It's just based on increasing the centrifugal force in a stepwise manner. So that is the major difference over here. And differential gradation, the centrifugation does not utilize any kind of density gradient. It is only based on the increasing speed. The different centrifugation speeds often create separation, maybe not more than 2 or 3 fractions. So the supernatant has to be further taken. Additionally, it has to be taken for centrifugation. With each step, the centrifugation speed has to be increased so that we get the desired particles. That is the only difference here. The advantage that density gradient has is that in density gradient, because we are creating a density difference, usually with one centrifugation speed, you can achieve your purpose. Unlike in case of differential, where you have to keep increasing the speeds. So this was about ultracentrifugation, one of the methods for isolating or purifying the viral particles. The other method that we have is ultrafiltration. Now this is a simple method, a much more advantageous method, because here we are carrying out the entire process under ambient conditions. No damage to the viral particle, no heat generation. So your damage to the viruses is highly limited. That is the reason ultrafiltration is usually used as a preliminary step for separation of the viruses. Now what we do here is the viruses are separated from this mixture, which may have different size particles, by using a filter which has a very small pore size that can retain the larger particles, but it allows the viruses to pass through. So when we pass the particles through the filter, as you can see over here, when we pass the solution through the filter, only the larger particles are retained, whereas the viruses which are smaller in size are going to pass through. So the viruses are obtained in the filtrate, which can be then purified or which can be used for study of the viruses. Now for this, we need to use a filter which has an extremely small pore size. So usually the filters which are used are made up of cellulose acetate or polypropylene, polysulfone, polyether sulfone. These are the different materials that are used for, or colloidal membranes. These are the ones which are used for making the filter that has to be used for viral separation. Now, in ultrafiltration, we can even use pressure-driven membrane filtration. So that also can be used. That is membrane filtration and ultrafiltration membranes, which are having pressure that will aid the process of ultrafiltration. Now here the ultrafiltration has a pore size of approximately 0.002 to zero point 0.1 microns, and a molecular weight cutoff, that is MWCO, molecular weight cutoff of approximately 10,000 to 1 lakh Daltons. So this is the range in which we can separate out the viruses. This was the second method of purification of viruses, that is ultrafiltration. The third one is precipitation. Now, precipitation is also called as crystallization, and it is one of the most effective method of obtaining viruses in the pure form. Usually, precipitation is done after an initial purification method or initial purification step using ultrafiltration or ultracentrifugation. So, in this, we use certain chemicals like ethanol or ammonium sulfate, which are added to saturation levels, and they are helping us to extract the viruses. So the virus has to be precipitated, and that is done using these chemicals, calcium phosphate or aluminium sulfate, or we could even use polyethylene glycol, that is PEG, a very common material that is used for precipitation. So amongst all of these, we have seen that PEG precipitation gives us a very good yield. It can in fact give us much more yield than even an ultracentrifugation process. So here what we do is we add the chemical in, or we add the salt up to the saturated levels into the medium, and then when we do centrifugation, when you add the polyethylene glycol or the precipitin, and then you do the centrifugation, we can see that the virus can come out of the solution. That is what you say, or that is what you call as precipitation. It is coming out of the solution. And how is this happening? It is because these salts or this polyethylene glycol is altering the solubility of the virus. So this is called as the salting out effect. It is altering the solubility. Now the virus is no more in the solution. It comes out of the solution, and this is what has been shown over here in this diagram. So this is the meaning of precipitation, one of the most chosen techniques for purification of viruses. The last method is chromatography. So chromatography, as we all know, is a technique which is commonly used for separating a mixture of particles, and this is based on the principle that viruses are going to get stuck onto or adsorbed onto the surface of certain membranes. So you use a solid or a porous membrane or a substance, and the virus will get stuck onto or adsorbed onto the surface of that substance. Later you can extract the virus. So ion exchange is one of the commonly used chromatographic techniques. You can go for adsorption, we can go for affinity chromatography, size exclusion chromatography. There are different types. But ion exchange chromatography is one of the widely used mechanisms for even the separation of viral particles and even many other biomolecules. What this is based on is the charge. All the charge based virus interactions are dependent on the difference between the isoelectric point of the virus and the charge on the surface matrix. So once you have selected the matrix in such a way that it has the opposite charge of the virus, when you pass the viral solution, the virus will stick onto the surface of that particle because they are oppositely charged. Now, the pH of the solution that is added is chosen such that the virus will become negatively charged and the resin or the matrix will become positively charged, or vice versa. So, they should have opposite charges. That is what we are looking for. And how do you make sure that happens? The solution's pH has to be maintained accordingly. Now, once they both stick with each other, the resin or the matrix and the virus stick with each other, now you need to use some kind of a material like an elution buffer, which can be made up of magnesium chloride or sodium chloride, or you can even alter the pH, due to which the viral particles which were stuck onto the resin or onto the matrix will now get eluted out. They will come out of the solution. So what we are doing here is, first we are making the virus stick onto the matrix and then we are making it come out of the matrix. So whatever solution we get at the end will be containing only the viral particles. Now the small molecules will take much longer time to come through that. So that is why we use size exclusion chromatography. But like I said earlier, we can even go for affinity chromatography, where we are going to use a particle that is very, very specific for the viral antigen. So we can use an antibody, make it stick together. But ultimately, what are we doing? We are taking resin or matrix in the chromatographic column, making that stick to the virus and then making that unstick to the virus. This is the simple way of explaining chromatography. So chromatography that can be used commonly is ion exchange chromatography, but we can also go for affinity or size exclusion chromatography. So these are some of the methods that can be used for purification of viruses. Each and every method has its own advantages and disadvantages. In the next class, we will look into what are the different methods for counting the viruses. Now that we have grown them and we have purified them, how do we actually measure our viral suspension? So see you all in the next class. Hope this was useful for you all. Thank you.